Good morning. Thank you very much, Bogumil, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you, Kate, and thank you for the invitation. It's really great to be part of this series. So I am assistant professor at Western University, and I guess my journey started, I always love working with plants. So for my PhD, I was very interested how certain plants, legume, form nodules that allow bacteria to colonize them and start the symbiosis. And this kind of affected me for life. I continue with the symbiosis. Now, at the very end of my PhD, I read a paper that was talking about genome transplantation and changing one species from one species to another. And I was so kind of interested in that in the end, I ended up working at Venter Institute for my postdoc and continue working with the team on the synthetic cell. So I was influenced by symbiosis in my training and also at the synthetic cell. And that's why you will see my research now kind of continues with that. So today, I will talk about few things that we do that are related to created microbes driven by synthetic genomes, and some of the work that we are starting towards creating novel organelles. Okay, so I know that this Build a Cell meeting. Often we talk about creating cell from scratch, all the cell components. Today it's going to be a lot about genomes, installing synthetic genomes, so kind of like the synthetic cell at Venter Institute. I do think, and that's probably not just me, but some people too, you don't have to agree with this, but writing and installing synthetic genome for me is one of the most important technology ever developed. And the way I think about it is that this is just the first technology that's going to allow us to change not only things that is around us, so all the nature, all the birds, all the trees, but ourselves. So how we going to look in the future, that's going to be kind of interesting. And that's actually Western University on the image. Okay, so we have different projects in the lab, but today I'm going to go over two projects briefly. One of them is what we call synthetic diatom project, and then I will go to some synthetic cells, synthetic organelles that we try to develop. Okay, so starting with the synthetic diatom project, I have to go back to Craig Venter, and when the first cell was created in 2010. This is actually the time that I just joined Venter Institute. It was about 3 months before the cell was created, so I really only witnessed its birth. I had no contribution, but then I had a chance to work with the team on creating of the JCVI Sen 3.0, which was the cell that was minimized. The genome was minimized almost by half, and the minimal cell was created. But 2010 marked the beginning of creation of synthetic cell. That was really the study that inspired, I think, a lot of people to start creating life driven by synthetic genomes. Now, here is a recent review that was published just last year, just showing some of the synthetic projects that are going on. So we're going back to 2000 when first some cDNA was synthesized, bacteriophages. Then we go to the Mycoplasma genitalium and mycoides, which form the basis of the synthetic cell. There were also a number of synthetic yeast chromosomes created, and E. coli, etc., and more yeast chromosomes. I was happy to see that our proposed SynDiatom project already made it on this map, so we are kind of right there. And obviously there are other projects to create synthetic mouse and synthetic human. Now, synthetic diatom project, we wrote little review or kind of perspective paper if you are interested to check it out. But essentially, for most of it, we're going to be focusing on Pheodactylum tricornutum. And we also have a X account and SID diatoms. We probably could be a little bit more active there, but if you are interested, just follow us. So Pheodactylum tricornutum, or PT, as I may be calling throughout the presentation, it has 25 chromosomes. The whole genome, nuclear genome, is about 28 megabases, which is a bit more than 2 times of that of yeast, Saccharomyces cerevisiae. It also has mitochondria, with the genomes are 77 kb and chloroplast 117 kb. One thing that I want to mention here is my student Emma Walker. She is current PhD student. She created most of those beautiful drawings and images, and most of the slides related to the chloroplast work they're going to be showing. So I just want to acknowledge that. They make these presentations much nicer. But those are essentially the three genomes that we have, right? The nuclear genome, chloroplast, and mitochondria. So if one day we want to create synthetic diatom or diatom driven by synthetic genome, we have to be able to manipulate all three types of genomes. So towards that end, to create this PT Syn 1.0, there are kind of three requirements. There will be requirements really for any species. So you have to have complete genome, you have to have a really good way to deliver DNA, and you have to be able also to clone it, your genomes, your chromosomes in host organisms. That is really important because they're going to be most likely too large to keep them in test tube. When you go to one megabase, the DNA is really subjected to all kind of shear forces, gonna start breaking apart. So you need to host them, especially when you build them. So in 2022, one of our student, he was a Dr. Daniel Rigier, he was PhD student in my colleague here at Western. He resequenced the genome of this diatom. At that point, we had 33 scaffolds. They were not completed. Some of them were telomere to telomere, but not all. And then he resequenced that, and now we have complete map of 25 chromosome telomere to telomere. When I was still at the Venter Institute, I developed this DNA transfer to this diatom that use conjugation. So essentially, using bacterial conjugation, we can move DNA to this diatom. Now, any other method at the time would give us very few colonies, maybe a couple. With this method, as you can see, this is one transformation plated here as this heart. We're getting essentially thousands of colonies. And conjugation can deliver very large fragments of DNA and sometimes whole chromosomes. So in theory, we should be able to deliver 500 to a megabase chromosomes directly from E. coli. The next part was, can we clone whole chromosomes? So similarly to what was done with mycoplasma for the synthetic cell, can we clone whole chromosome? So starting with one of the smallest one, so this one is chromosome 24, at that time scaffold 24, which is approximately 500 kb. I attempted to clone it in yeast. So in yeast, we can do this transformation associated recombination when you have your plasmid that you just have, you open that plasmid, this vector, you will have homology ends. They can be anywhere from 50 base pairs to a couple kb if you wish to. And you position them close to the end of the DNA that you want to clone it. So it could be part of the chromosomes or the whole chromosome. When I try to clone the whole chromosomes, it always fails. And it is kind of common knowledge, or at least that some of the fragments or some DNA cannot be cloned in yeast because maybe they are too repetitive. The DNA will fall apart, recombine in yeast because yeast has such a good homologous recombination. Now, this is not necessarily the case with everything because yeast also needs special origin of applications, at least every 50 to 100 kb. So even we couldn't clone the whole chromosome, the chromosome was cloned as fragment, 100 kb fragment. So those are individual fragments were cloned. And then later, when they were separated in different yeast or E. coli strain, we added extra origin of replication to fragment 2, 3, 4, etc. And then when we tried to reassemble this again, it worked. So we actually could clone whole chromosome as long as we're adding those origin of replication. So we have now those rules that if the GCDNA, if the content of DNA is more than 40%, you will need to add extra origin every 100 kb. And I just want to know that this origin is the same. We have this repetitive sequence 3 times plus 4 times on the plasmid, and it's still stable in yeast. So you can have repetitive elements in yeast as long as there is enough origin of replication. Here is just showing that this 500 kb chromosomes could be moved from yeast to E. coli. And then later on the right side, you have pulse field gel showing that, yes, indeed, looking at 4 E. coli colonies, we have approximately 500 kb band. And on the left side, you see a yeast genomic DNA marker. So those are essentially yeast chromosomes. So we could have them. We can have whole chromosomes in yeast or E. coli. So that was good. The next thing was, can we clone the chloroplast and mitochondria genome? They are very low GC content, and they potentially could cause problem in E. coli. And we knew that because when we were working with the synthetic cell, which has DNA closer to 25% GC content, if you want to put fragment of DNA more than 10 kb in E. coli, they just fall apart. So essentially the question was, if you have closer to 30%, could we maintain it in E. coli? The prediction was they would be great for yeast because they're below 40%. So cloning in yeast is going to be simple, but could you clone them in E. coli? So here is Dr. Ryan Cochrane. He was a PhD student in my lab. He graduated now a few years ago, and he started working with mitochondria. So he had two approaches. He did PCR approach when he cloned minimized version of the mitochondria genome. So minimized because essentially he removed or he skipped in this approach a repetitive region that was about 30 kb. So by just doing PCR and assembling it inside this, he was able to get that. And in the second approach, he did the star cloning. When he designed the vector, this vector actually was split into two. And then each one has, like at least each of those fragments had one homology end to the linearized mitochondria genome. So he used that TAR cloning to also capture the whole genome. So in the end, we had the whole genome captured by TAR cloning or by using PCR-based method that they were cloning yeast and then later in E. coli. So that was good. At that point, Emma also joined this project with cloning of the genomes, and Emma was focusing on chloroplast genome. So based on everything that we already learned with the mitochondria, Emma quickly designed PCR-based approach to do the very similar kind of strategy, just PCR them, PCR some cloning vector, and then assemble them in yeast, move to E. coli. Now, we obviously can synthesize them, but PCR is still cheaper. If you want to just initially just test it, can you maintain it? So we still recommend that method. Now, in addition to the PCR, as we are thinking of creating many versions, so going to the typical design, build, test, learn, synbio cycle, Emma created a slightly modified, version when we have pre-clone fragments. And I'm just gonna go through this into a little bit details. So to pre-clone it, Emma first design strategy to essentially domesticate this chloroplast genome. So we wanted to use the same site to release every pre-clone fragment, and the enzyme was sub1. So essentially, she had to remove all the restriction site, sub1 restriction site into this genome. So she did that by including them in PCR, and those are the fragments that she did. Eventually, she created nice 1/8 genomes that they can be open up with a single restriction enzyme, as I said, the SAP1. Then you can add, so that's going to release the initial cloning backbone. Then she has another one cloning backbone that will have overlaps to two regions, two fragments. And essentially, in one tube, you could do later the whole assembly. So you perform either individual digest and you check them, or you can just put them in one tube, as I mentioned, and digest them together. Once you digest them, you throw them into yeast. Now, you may ask, why are we not doing golden gate assembly in vitro? Potentially, you could do that. I do notice that as you start working with larger fragments, we kind of combine this when it's almost partially golden gate, meaning that we domesticate everything, release with one enzyme, but we have larger overlaps and actually do the assembly inside yeast. That I think is more foolproof method to create chromosomes that are larger than 100 kb. And then when she did that assembly, it was quite amazing. So it was one test tube. She did many other ones, if you can go to the paper and check it out, but I just showed this one example. When she did just in one test tube, so throw all the fragments plus the recipient plasmid, quick digest, throw it inside yeast, and here picking up 10 yeast colonies, they were all perfect as screened by multiplex. So essentially, Emma has this multiplex PCR design at the critical junctions that overlap the junction, and we screen them with the junctions, and they all look really good. And you see the negative control and positive control, as well as the ladder. So taking some of these colonies were moved to E. coli, and in this particular case, there were 3 colonies, colony 1 and colony 3, moved to E. coli, and then 3 colonies in E. coli screened for each, and they're again, they're perfect. So this is really good. This is really good as we start building all kind of new version that this works. This is such a robust way. And when we sequence, those genomes are pretty good. We do notice that if you use initially PCR, we are using Takara GXL's enzyme for most PCRs. We get approximately 1 mutation in 10 kb. But if you pre-clone the fragments and you pick up the good ones, the perfect one, then you shouldn't really be getting extra mutations later on. So that was quite nice. Now, are they do they have any effect on yeast or E. coli? Here is just showing you E. coli assay when we test the growth. Now, what I'm gonna mention, almost everything that we do in the lab, we are cloning enobacterial artificial chromosomes that we can keep it at very low copy number, so essentially 1 copy, or it can be induced with arabinose to maybe 20, 30 copies. So that's how it's all set it up. So here, when we have just with chloromphenicol, so we are selecting for the plasmid, but they are low copy, they are not induced. Essentially, everything grows the same. And the controls are the PDMI 7-8 and then PDMI 2 SAP, so the blue and green. They behave very similarly to the one that contains actually whole genomes clone, chloroplast genome. Now, if you induce it, to have higher copy number, you start seeing that the one that carry the chloroplast genome, they grow a little bit slower, but the E. coli is still growing, so it doesn't really stop. So, that's good. If you want to just isolate DNA at the very end, you can do it for a couple hours to induce to get higher copy number. We did a couple other tests. So another test is what happens to the genome if you grow it to, even if the E. coli grows, but do you start having more deletions or some kind of rearrangement? So this is going through 60 generations. When generation 0 refers to taking them directly out of yeast and screening right away. And this is again with the multiplex screening at 6 locations. We don't really see any like gross rearrangement. And after 60 generations, that was the same case. So once again, that was good. And some of those clones were also sequenced. Now, I'm gonna just kind of bring your attention here that the chloroplast genome has 32% GC content. We did the same test for the mitochondria, and it wasn't, we didn't get the same results. So essentially what happened, we did clone mitochondria from PT, but we also clone mitochondria from another algae, another diatom, Pterostoceras pseudonana, which the GC content is even lower. So the GC content is 30%. So when we move this when we move this mitochondria out of yeast and check it, E. coli colonies right away, which is the generation 0, what we call, essentially they were all good. Again, testing with the multiplex throughout the genome. But after 60 generations, we noticed that there were at least 4 of them that lost at least 1 fragment. And some of them, in different locations. So we start seeing some deletions. So again, as we get in close to 30%, with this particular diatom, we can still kind of quickly work it, but in long term, they're gonna have problems. Now, with PT mitochondria, we did not see any problems. This one has GC of 35%, so it's even higher than the chloroplast. So something to keep in mind as you start adopting this method to other organisms. Now, how are we gonna deliver? So essentially, I quickly when I showed you that we can really clone all the genomes in yeast, and then we move them to E. coli. So we are really ready to start creating synthetic chromosomes. But how are we gonna deliver them? The Venter Institute team kind of came with this cool method of genome transplantation. We really cannot do genome transplantation in the species, at least not yet. So one of them will be potentially the conjugation. we're hoping that 500 kb, since we can clone it in E. coli, we probably can move it. We probably can move it into algae by conjugation. So we would have this in E. coli, we would conjugate to this algae, and then we would have it as a initially circular chromosomes, but we could engineer it to potentially release the plasmid vector backbone and have it linear chromosome. Another method that may be gonna work is gonna be electroporation. So electroporation in this algae wasn't working that great. People are using a little bit over the last couple of years, but you get some colonies. So if you wanna just move and get few transformants with small plasmid, that was fine, but it wasn't working great. So you could not imagine that whole chromosomes, we would be able to move it with such a low frequency. Just last year, Emma here came also with this idea that if she protoplased those diatoms, protoplast method already existed, but no one really tried to use the protoplasted cells with electroporation. And she did that, and the results were absolutely, like, I was just amazed. So when we look here at this particular cells that have no protoplasting, so those are the one without the alkylase. Alkylase are the chemical that we use for protoplasting. You see that that there are a certain number of colonies we get with electroporation, but as we protoplast it, we get much more. We get thousands comparing to 50. So that is huge improvement. Also in the past, most of the protocol called for micrograms of DNA, but Emma could get even with 1 nanogram of a 10 kb plasmid that could be delivered. So this is brand new. We have that in preprint, and we're gonna be, so the full manuscript hopefully is gonna come sometimes this year with some additional data. But now we have a new way to potentially install them. Now, what is really also interesting with this new electroporation method, Emma also showed that we can assemble DNA right in this algae. So anywhere from 2 fragments to 3 or 4 even fragments, we can assemble the DNA. So maybe we can start building chromosomes inside this algae. So transforming smaller pieces, kind of like what was done for anyone that is familiar with Saccharomyces cerevisiae 2.0, how it was created by building inside the yeast actually from larger chunks. So maybe we can do that in this algae too. Okay, so when are we gonna have our first synthetic diatom? Hopefully really soon. We have this preliminary work. I know that we are a very small lab here in Canada, so we can't do this alone. So that's why I started this. I started those workshops. That essentially those are only couple hours workshops. We had 6 of them. That maybe twice a year we meet. And we just discuss, what grants we can apply, what can we do, what are the next steps. We have another one coming up on February 26th. If anyone is interested, just let me know and I will add you to the email. And those are really fun things because we're talking about specific diatoms, but we're really talking about creating synthetic organisms. So many of the rules that they're gonna apply for those diatoms gonna apply for other eukaryotic cells or even bacteria. So you are definitely welcome to join. Just let me know. And that was it for the algae. Now I will switch gears a little bit and talk about synthetic organelles. So a few years back, actually that was during the COVID era, a few of my students, we kind of wrote this perspective thing as the synthetic organelle field start to emerge. And the whole goal is to essentially be able to turn any bacteria into novel organelles. So could we do something with the molecules or rhizobia or cyanobacteria, and could we put them inside these or maybe mammalian cells or plant cell, and then later create new functions that never existed before. Now, for me, this all started when I was at Venter Institute still, and we had to move genomes from bacteria to yeast to start engineering them. And at that point, one of the kind of idea was, could we simply just mix bacteria that already has all the yeast elements inserted that could replicate in yeast? Instead of just isolating the genome, can we just mix them with these protoplasm and add some PEG, so polyethylene glycol, in a very similar manner that you would do with DNA transfer? And could the DNA be transferred to yeast? And that worked on the first trial. So those are just few of those experiments in my life that you just do this first time and it works, and I was just amazed. So the question was later, how the DNA transfer? So we had the bacteria and we have the yeast. The yeast is spheroplasted, so some of the cell wall is removed. But is it that the bacteria fuses at the membrane? Is the bacteria simply being, broken, it just degraded in the PEG solution, and then the genome is released and then taken up, just as a would be normal DNA transfer? Or is the bacteria engulfed, and later, later the bacteria is getting degraded and goes to the nucleus, or somehow the bacteria provides something to it that it can survive? So there were, few papers that we had, but essentially one of them was pointing that, so those are TEM pictures pointing out that it may be actually the pathway 3A. So essentially what you see here in A, those are yeast spheroplast. So this is yeast, nicely spheroplasted. Here is mycoplasma. So those are those mycoplasma cells. And here you have protoplasted yeast and mycoplasma cells. And you start seeing them. We kind of start observing different stages of self-engulfment. So here they were just attached. Here you see that the yeast kind of starting to get engulfed even more. And then we kind of seen something like the mycoplasma was sitting inside the cells. So that was kind of the showing that the bacteria goes most likely through this route, and then somehow the DNA is getting extracted, and the DNA survives while the whole cell is really taken apart. So since essentially that time, I had a dream that maybe we can take this and turn it into organelles one day. Can we stop this process? So here is just this kind of outline that we put in 2020. Later on, there were some papers that start also kind of going with the same idea. Can we start turning different bacteria like E. coli into organelles? So a few papers came from Peter Schultz's lab. I think that's like 2018, 2019, when they were showing that they could establish first level of such a symbiosis. So for me, that was really exciting because it shows that it wasn't really impossible dream, but people are making some kind of first steps, and maybe we can actually get this process going, that we will be able to turn any bacteria into organelles. So here is kind of two designs we could imagine. So essentially, if we could predict and design and add all the genes to the bacteria that would allow it later to be engulfed by yeast, provide some benefit for yeast, without also and prevent the yeast from degrading the bacteria, then we establish this symbiosis, right? Now, this is not going to be so simple. So we can have also design tool where, in addition to this, what we think are the survival genes, we could also add genome survival elements. So essentially, those are the elements that I kind of showed you in the past when we want to adjust the genome to go and survive. So when you do the fusion, if you engineer this properly, you may end up with the symbiont living inside, but if you don't, the yeast is still taking the bacteria apart, you at least end up with the genome inside the nucleus. So you at least have some kind of indication that the engulfment is happening, and then you have to continue, going, depends what is your kind of desire. Is the desire to create the organelles or just have the genome? So those are just some kind of the elements that we would have to engineer. Now, in our lab, we started with 3 different organisms that we proposed for synthetic organelles. Initially, we wanted to have work with the mycoplasma, but unfortunately in Canada, mycoplasma is considered biosafety level 3, so we really couldn't do it. But we have replacement for it, another molecule, E. coli plasma Leitlawi, and this one is mostly all the work is done by Daniel Nusipora, who's PhD candidate. Then we have few rhizobia species because we would like to create nitrogen-fixing organelles. So when we started this project, there was really no nitrogen-fixing organelles other than you probably all know about bacteria rhizobia that goes and colonize plant roots. Again, takes me back to my PhD research and forms almost organelles like. But in the last few years, there was a lot of research identifying those algae cells that can live in diatoms and fix nitrogen. So we have those nitroplasts identified. But still, we would like to take really rhizobia and turn into novel organelles that fix nitrogen inside plant cells or any cell, not just nodules, but also any plant like rice and wheat and corn, etc. But also in other organisms. So this is research being pursued by Jordan Mini, another PhD candidate, and then we have Samir, who is working with Haemophilus influenza as another potential organism for creating organelles. As well, we consider them all for genome transplantation. So one of the rhizobia species is Cynorhizobium meliloti. This one is interesting. It has very high GC content. Its genome is split into three essentially main chromosomes. There is another one, what is called chromate, because it has some essential gene, PSMB, and large megaplasmid PSMA. This PSMA plasmid mostly has those symbiotic genes. Now, it was previous research done that was actually done even before, well, actually soon after the synthetic cell was created, but before the minimal cell was created, that couple science, George DiCenzo and his team here actually in Canada, they were able to remove those two plasmids and show that the bacteria is still alive. So they essentially reduced this genome by 45%, and only some of the essential genes were moved on the main chromosomes. Now, when we saw that, we said, well, this is such a great way to potentially start engineering this genome for our synbio purposes. So first of all, we would use those two origin of replications from those from this chromate and the megaplasmid, and created our designer plasmids, and that was work by Stephanie Brumwell, who graduated from my lab. That was actually my first PhD student, my first student that started in the lab. So she did a great job here, and then so she developed all these genetic toolboxes for this rhizobia, and that would allow us to start engineering them and slowly either turning them into donor recipient cells for genome transplantation or for the synthetic organelles. Now, more recently, Jordan took some of the tools that were developed for Cynorhizobium meliloti and start testing them and developing new tools for other bacteria, other rhizobia. So the reason that we wanted to go to other rhizobia, because Cynorhizobium meliloti mostly fixes nitrogen when they are present inside the nodule. But we want to have also rhizobia that fixes nitrogen that they are living outside of the plant. And we just wanted to see, can we start moving all those tools? And yes, we could, quite a few of those tools, plasmids could work in different rhizobia species. So now we have series of rhizobia that we can start converting into the nitrogen fixing organelles. So that is exciting. And we can start adding now fluorescent genes, etc. So for example, here is the Cynorhizobium meliloti. Here's I favor these VL648N, and here you have a picture showing that just mixing them next to each other, you have the rhizobia in red and yeast in green, and we are just testing different engulfment strategies and looking for survival to get to our syn organelles, synthetic nitrogen fixing organelles. So that was just briefly about the one species. Now I'm gonna switch to E. coli plasma. So E. coli plasma is another very interesting one. So the reason we want to choose E. coli plasma, I mentioned briefly, it is molecular, so it has small genome, a little bit bigger than Mycoplasma mycoides, the one that was used for synthetic cell. So E. coli plasma is approximately 1.5 megabase versus the 1.1 megabase, but it also has standard genetic code. So in that sense, it's different from those spheroplasmas that are usually used for the genome transplantation. E. coli plasma are also related to plant pathogens, phytoplasmas. Phytoplasmas are they have very small genome, like 0.6 to 1 megabase, so very tiny, but they really cannot be grown in the lab. So by developing some of the tools for E. coli plasma, hopefully we could also like help engineering phytoplasmas. And phytoplasmas affect a lot of plants. They just destroyed flowers and really decrease the crop. So, you know, we are interested for synthetic biology, but there could be important agriculture application here. So starting with the E. coli plasma. Now, so this work is, as I done earlier, mostly done by Daniel. And Daniel had here a lot of, he's extremely patient because I started some of that work with the E. coli plasma when I was at Venture Institute and I failed completely. I could not transform it even once. It was not a fun microbe to work with. But in the end, Daniel has now 2 microbes that he started working with. So there is this E. coli plasma Leigh-Lowy, with a lot of research was done, but not much genetic transformation, but just looking at the membranes and really evaluating the microbe, but not necessarily developing a lot of genetic tools. We can clone that genome inside this, but again, transformation was really not good. So Daniel got another strain, Leigh-Lowy 8195, which is very similar, but has no restriction systems. So therefore, it was good candidate for developing some genetic tools. And again, if we succeed with genome transplantation, we're hoping that the PG88 would be donor and the other one is recipient. So I'm not gonna go to this in details, but Daniel developed a series of plasmids, developed electroporation method that now we can load all kind of different genetic tools and propagate them and start engineering. He also did a small experiment that essentially we just try to see, can we improve the genome transfer to this microbe? So he performed this experiment when he would transfer plasmid, cure it, transfer another plasmid, etc. Went through 4 generations, retransforming the strain. Then he screened some single E. coli plasma strains, and he actually discovered strain that is much better with DNA uptake, and he has now a strain that is about 200 times more susceptible to like DNA transfer, so it's much better for DNA transfer. So again, for genome transplantation and really any synthetic biology work, it's much easier to work with. We have now deposited that strain to ATCC, so anyone can get that. So that was really good that we could do that. Now, this particular strain had also no recombination, so Daniel also reinstalled recombination, and I'm not going to go into details into this diagram. Daniel is preparing manuscripts, but essentially he restored recombination in the strain that we have this DNAE, so you can start now installing your pathways, whatever you want to do inside it. Finally, he also was able to install whole yeast, artificial chromosomes using that method, and we can move the genome by cell fusion to yeast. So again, we are at the same point as we were with mycoplasma, that we can fuse the whole cell with these and move it, so we know that the engulfment happens, and can we stop it at some point and develop into organelles. And here is just confirmation that when we fuse it and we have those resulting yeast colonies, they are not just some kind of fake yeast colonies, spontaneous, colonies that, but they do actually have E. coli plasma in them. And we just check them quickly by multiplex, like we check as a first screen. Now, it wasn't really easy to clone that because this E. coli plasma had toxic genes and so on. Anyways, Daniel is preparing this nice manuscript, and you can read that about this very soon. Finally, I know I'm jumping a lot here, but I just wanted to introduce you to all those different 3 species that we're working, because maybe one of you gonna be really interested to work with us on all of them. And I just want to show you where we are with each, because most of the genetic tools that we developed, they were developed in the last year or 2. So the final one here is Haemophilus influenza. This one was special because this is the strain that Dr. Hamilton Smith used to identify, the first restriction enzyme, HinD2. And we also try to fuse this strain with yeast, and it worked. The whole genome can be moved by cell fusion, but really it fuses much better when you remove the restriction enzyme. So this strain has two restriction systems. If you remove them both, you can about 250 transferred versus the wild type. And we've done that with other species too. We know that if the bacteria has restriction systems, the fusion to yeast is not great, so you have to remove them. But so now we have essentially all those species that we can now transfer them to yeast, engulf them, and we know that this works. So here is an image that we capture still during that work, showing that yeast protoplast or spheroplast, and showing that the bacteria was engulfed here in some kind of vacuole. So that was our Haemophilus influenza. So we know that this happens. We know that after a couple hours, we still have the bacteria inside, but can we maintain it for much longer? So since then, this was just research published last year by Samir, who is a PhD candidate in my lab too. And Samir developed a lot of genetic tools. If you start looking at Haemophilus influenza wild-type strain, most people use natural transformation, which works to transfer DNA, but it's not great. But when we use this, restriction minus strain, we notice that transformation rate improved greatly. So I'm just going to walk you through this image here. What was also interesting to see that on the negative plate, even without any DNA, when we try to transform it, the wild type forms this biofilm, and our restriction minus strain doesn't have this biofilm. So this is something to be investigated further. Maybe someone is looking into that. We could definitely partner on this. But yeah, wild type has this biofilm, and if you remove the restriction system somehow, because when we remove the restriction systems, we also remove the methylase and the nuclease. So obviously the methylate many sites, and we still have to see what genes were upregulated or downregulated to cause that. But no matter what, at least for the DNA transfer, you see that if you move this particular plasmid, this is small plasmid, we're getting very few colonies here. They're kind of hidden in this biofilm, but with the restriction minus, we're getting tons, hundreds, and then this is different plasmid. And once again, we get lots of colonies here versus very few with the wild type. So we can really easily engineer this for any purpose. We have series of plasmids developed with different antibiotic selections, different visual markers. I'm showing you here, for example, GFP and RFP and some other markers. So we deposit everything to add gene as much as we can so everyone can share. So that is, we're hoping now that H-flu is going to become as simple to engineer as E. coli. It has also a very small genome, 1.8 megabase. So again, another good microbes to engineer for different purposes. And this was the very first genome to be sequenced of bacteria. So it has also very historic importance. Now, as we start taking all these bacteria, so I showed you that for all of them, we develop all these genetic toolboxes so we can really start engineering them and creating those dependencies. Some of the initial things that we also do, we try to establish the co-culturing, conditions. So how are we going to culture them? So for example, Haemophilus influenza is oxotrophic, requires addition of hemine and NAD. Yeast you grow on different completely media. So we try to figure out the conditions that we can grow them together, right? The temperatures. And one of the interesting things that Samir here discovered is, for example, if we use yeast that is oxotrophic and essentially requires adenine addition, the yeast essentially without adenine turns red. But so I will just show you here essentially kind of like in these conditions here, if you see my pointer. So this is on the yeast loaded. So yeast is required, it's going to be red without the adenine. Unless it's supplemented, as on the last one, the yeast is white. So that's the phenotype. Hopefully, many of you are aware of this and use that in the research. You can kind of screen that by the color. Now, what we notice that if you start co-culturing the yeast with our H-flu, so H-flu is always 100% at the top, and then we have some different combinations of yeast and the H-flu. As you can see, we kept the yeast constant and diluting the H-flu. You could see that the yeast complemented, the H-flu complemented the yeast, so the yeast became white. So the yeast was able to provide some of this adenine that was missing. And we had a couple different conditions when 10% adenine was added. Actually, this looks even nicer. When 10% adenine is added, the yeast seems to be grow initially faster, but still suffer. But if you add the bacteria, then the bacteria can complement it. So anyways, this is not really engineer symbiosis yet, but we see some interactions here. And our goal is not to have them co-culturing, but eventually have them inside. But we do need to evaluate all the conditions. And that's what we do with all the microbes. We try to figure it out, how to grow them together, what are going to be the conditions when one of them grows on it or both of them grows, and do they have any negative impact on each other or beneficial impact? Like in this particular case, it is beneficial impact that we just kind of by accident discover. So that's where we are. We have the species, we have the genetic toolbox, and the engineering will be just starting now. And the whole point is right now that as we do the engulfment, which we can do with all the species, can we essentially inhibit this pathway here when the bacteria is getting engulfed and turn them into the organelles? Because really, if we can do that, then we're going to be able to create almost any organelles in the future that we want. So that's the vision, that's the hope. I will stop here because I talk about quite a few different things. And I can answer any questions or meet some other time. But before I go there, just a few things I want to thanks my lab. I had amazing time. I'm 7 years now at Western, and this is just a picture we took a couple days ago. Many students are really not here that graduated. So the success of all this work was done by everyone, from past postdocs, graduate students, undergrads, and all the collaborators that we work with over the last 7 years. And I listed all the great granting agencies. Now, I have applied for a number of different projects, proposals. I hope to have some positions open. The one, I may have 1 or 2 PhD positions for genome transplantations. We want to develop genome transplantation for E. coli plasma, Haemophilus, Deinococcus, also radiodurans. We will have hopefully more positions for the synthetic diatom project. I am getting answer for some grants in the next 4-8 weeks. So if anyone is really interested in any of the work I presented for students, please let me know. We'll be happy to have you in our lab. Also for collaborators, if we have always this open call, we can work with, obviously many of you on different grants, but we have this open grant that if you're ever applying for a grant and you think that we could work together, just mention us in your grant. And if you get your grant, we can apply for additional $300,000. So if any of this is what I show kind of relevant to your work, we could apply for this ever. But unfortunately, you first have to get your grant before I can apply. So that's the catch here. But this is open grant, one opportunity. Finally, we're gonna have pleasure to bring Build a Cell to Canada. I am really, really happy. The Build a Cell meeting workshop number 14 will be here at Western on May 2nd. Now, most of those pictures are probably June, July, so I'm not sure if it's going to be so green. Actually, one of them is in the fall. You see that the trees are kind of turning color, but hopefully, winter will be over by then. So I welcome you all here to come to Western, and should be great workshop. So thank you, and I will just take questions now.
Thank you so much, Bogumil. That was really awesome. There is a lot of questions in chat. Can you see chat?
I can see chat. I will just open it now, and okay.
If you can read a question out loud before answering,
Sure.
that's going to help with the recording.
I see that you were scared. Okay. Symbiosis is the coolest thing in nature. Weird but cool. Good. I agree. I feel cobra chicken, so Canadian. Okay. Yeah, cobra. Yeah, those are cobra, right? Cobra chickens.
I think that was a comment when you were
I
showing
know
that
those
picture
geese.
full
Yeah.
of geese. Yeah.
Yeah. Cobra snakes. I don't know. There were different names for them. Okay, here's perhaps stupid questions, but why do you clone the chromosomes in yeast instead of cloning in E. coli? So essentially that depends on the size, but from everything that I've done, and I've done a lot of cloning when I was at Venture instead of coming here, and yeast is much more forgiving. So, and again, based on the size, if you wanna clone whole chromosomes like 500 kb, the question was maybe it's gonna fall apart in E. coli. Now, for example, if I was going to, if we are able to insert E. coli markers into like mitochondria or chloroplast inside the diatom, then we can isolate the DNA and move it to E. coli right away. We don't have to go to yeast. But if you're starting with new project, I still recommend to cloning in yeast, but there are the rules. If you have anything above 40% GC, you will have to add extra origin. So go to E. coli. If you have anything 30% lower, go to yeast because things will not fall apart. So yeah, certain things you will be able to clone in E. coli, but again, those two holes are important. And then when we building synthetic chromosomes, we most likely gonna create maybe 5-10 kb pieces of DNA, and we can throw them in easy and assemble. I know we can also use variation of Gibson assembly to do certain fragments in E. coli. Now, prokaryotic DNA may be more toxic in E. coli than in yeast. So far, I closed 6 or 7 whole bacterial genomes inside these, and I did find a single toxic gene. So for example, and that was in E. coli plasma. So for example, we can clone the whole H. flu genome in yeast, but there was scientists in Canada was cloning H. flu in E. coli and only able to clone about 10%, and then it became toxic. So you have to look at this. So that's the question here. What is the size limit for a genome you could amplify in E. coli? Could it be as big as SYN 3.0 genome? You could amplify, you mean, I assume propagate in E. coli. So SYN 3.0, which is 500 kb approximately. Yeah, so we cloned this whole chromosome of we cloned the whole chromosomes of the chromosome 25 of algae, and that worked, which is essentially the same size of SYN 3.0. Now SYN 3.0 would never replicate in E. coli because of the GC content. Now, I know that some people talk about it in the past, and I know that certain genes were, the genetic code was changed from the non-standard to standard for mycoplasma. But could we resynthesize 3.0 to have higher GC content? That could kind of would be fun project. Resynthesize it and just can we go from the 25 to 35? Not much, maybe, but to 35, and then you could actually do it everything in E. coli most likely, unless they're toxic genes. So that's one of the reasons. So yeah, size is okay, but there could be toxicity. When you consider free-living organisms to turn into organelles, do you consider minimizing the genome of the free-living candidate first? Well, I mean, I would say you can, you could, not necessarily initially, because the problem is so big, but you could install something that will mutagenize the strain, right? So the way I kind of see it, as long as you establish any kind of symbiosis, that you can move the bacteria inside the yeast, and let's say you maintain it for a short period of time, even couple days or couple weeks, then you potentially can mutagenize the whole population of bacteria, and then so that way you kind of minimize it, and then you start evolving it. And I do think that once we do go through maybe 5-10 different bacteria and we understand the rules, then most likely you're going to be able to minimize them. Now, in terms of H. flu, the genome is already 1.8 megabase, which is like a third of E. coli. it is auxotrophic for few things. So yeah, we could potentially, that's actually our goal, to provide, for example, hemine from yeast, and then so this is partially already minimized for us, right? So you can imagine. And I know that there was research in Dr. Schultz's lab on E. coli minimizing some of the genes, so you could do that. Could you briefly discuss considerations for the host for organelles? How do you pick up a good recipient? I mean, really, we don't know. I started with yeast. We considering yeast, something that also obviously we have all these collections of knockout yeast. Uh, I look in nature. I try to be now, as we kind of try to propose this, I try to be inspired by nature. So for example, we know that legume plants have already symbiosis in nodule with this rhizobia. So could we use some of the legume plants already to start creating plant protoplasm and bacteria? Those are very difficult and challenging things, but you can start looking at both the donor and the recipient cells that already have some kind of symbiosis, or they can tolerate at least temporarily bacteria. So right, so that's how we do. But yeast was simple, and we saw the engulfment, when we tried the genome transfer. So that's why yeast is kind of obvious candidate. Do you ever consider completely artificial organelle made from synthetic cell? Absolutely, yeah. So I mean, the synthetic cell, but are we talking about the synthetic cell like Kate and everyone, one of you guys are doing, developing the membranes? So I think yes, absolutely, both cases. Creating those compartments, and so this is different type of research. I don't really do anything with it, but absolutely, that you make those compartments. And people are working on that. But it could also be referring to the synthetic cell, like the mycoplasma synthetic cell, right? I guess we call synthetic cell now when the cell is completely done from scratch, and in the other case, we always say cell driven by synthetic genome, right? As far as I understand. I understand the long-term vision, but we are the short-term advantages of engulfment synthetic versus co-culturing. What do you anticipate coming from this? So, for the co-culturing, like, yeah, people ask me, why do you do this co-culturing when you want to engulf it? Well, when we engulf it, even when we're going to be able to, we're going to engineer. So let's say the yeast, we take yeast that is auxotrophic for tryptophan. We overexpress that in our H-flu. And then we put them together and say living, and now the yeast survives. I want to be sure that whatever media I grow, the H-flu doesn't survive on the media, right? Because if they survive next to the yeast, it's kind of misleading. So we try to understand all the conditions when they grow together, when they don't grow together, and then I think I am not that smart. So as we start engineering the symbiosis, maybe we can partially engineer them and let later evolution to take care of it. So then we start having more and more stringent condition that the cell has to be engulfed. Evolution keeps helping us in the lab. With the coliplasma, as I mentioned, Daniel developed this better recipient strain. We had now some plasmids couldn't replicate. Using evolution, we actually evolved them to now have plasmids that are stable. I really love the combination of all the current knowledge with the evolution. So that kind of a little bit goes with the co-culturing, but we need to kind of know these conditions. Don't be scared, I'm a nice guy. What else? Could we please comment on the advantages of engineering of our organelles from scratch versus engineering existing like mitochondria to enable new functions? So yeah, we are, so a lot of people are engineering organelles from, existing organelles by multiple ways, right? Like you can express genes in the nucleus, transport them to to the organelle, or edit them, etc. And we are actually considering that too. But once again, I am this, from the kind of purest way, we want to have the ability to engineer everything from scratch. So if I want to make fully synthetic cell, that one day the cell may, so we want to recode the genome, or just do whatever. Maybe we're going to completely eliminate 90% of the DNA from chloroplasm and mitochondria and move it to the nucleus. you know, we don't want to play God, but we want to be like, whatever you call it, we want to have the ability to manipulate every organelle. And yes, engineering the existing things are exciting, but why not to, for example, engineer new organelles, right? I think that it is very hard, like the nitrogen fixing problem is forever, right? We are making advances in all kind of ways, moving pathways to ease. But then we have this rhizobia that already produces nitrogen. Why not to kind of turn it into organelle inside other cells, right? So I think everything is important, but both kind of strategies should be, you know, pursued. Is GC content the only key criteria, or are there other challenges that are more fun, like to be generalized? So I mean, GC content is only when you worry about the genome transfer. Obviously, well, sorry, that there is, you know, when it comes to genome transfer, that's GC content is only kind of plays any role. If you're gonna be doing organelle transfers, that shouldn't matter, right? Because the organelles are already different GC content. Like chloroplast is 32, and the main chromosomes in algae are, you know, 48, let's say, right, or something like that, 40 or 48. I'm forgetting right now. So for the new organelles, that doesn't matter. For the DNA transfer, it matters, but obviously, if you have more transcription, right? So if you're moving bacteria into bacteria hosts, you're gonna have more toxicity. Uh, there are kind of research done when trying to simply clone bacterial like libraries in bacteria, and certain genes could never be cloned. Or so like ribosomal proteins many times cannot be cloned in E. coli, for example, from other species. So yeah, you have to consider, right? So those are some of the other rules, right? Which host are you using, prokaryotic to eukaryotic? Now you can take advantage of different genetic codes, right? Like the mycoplasma has different genetic codes. So if we keep the same genetic code but change the GC content, we could move it to E. coli, and essentially every single protein almost that has tryptophan, that would be stop coded in E. coli, would not be properly translated, right? Would be terminated, truncated, so therefore they would not be toxic. So keep genetic code in mind, keep that, and right, and just those are the kind of solutions as you're moving between species, and then kind of rules to keep in mind. What are the size that could be transferred? What are the upper bounds of size that could be transferred? So, you know, for E. coli, I did transfer 500 kb by electroporation. I do think that by other methods you could do much larger. By cell fusion, there is essentially no limit. By cell fusion, I move 1.8 megabase to east, but then what we kind of are at least thinking, if you, let's say, if you were going to engineer mammalian cells, we know that we can fuse all these chromosomes to have approximately 12 megabases. And so in theory, you potentially can move 12 megabase. So yeah, if you do cell fusion, you could move much larger. Conjugation, I think 1 megabase probably will work. And as we develop in some kind of protoplasting method, people fuse sometimes protoplast like bacillus to bacillus. So yeah, it will depend on the method, but method and obviously what you're putting, right? As I said, right. But whatever you do for everyone, keep everything low low copy number. That will be the conclusion too. That much, because for example, the whole chromosomes for algae, low copy number was fine. If I induce it to high copy number, very quickly falls apart. So yeah, I think those are all the questions, unless someone wants to just ask questions in person.
I think you answered all the questions. Thank you so much. Yeah, I was really interested in when you were discussing the limits, because that's going to inform what are the functionalities that we're going to build from scratch, and that was really cool.
Yeah. So I'm gonna add that, the synthetic cell and the synthetic genomes, maybe some of you've seen, there is a new call, there was called that, from UK to create synthetic plants, whole synthetic chromosomes and chloroplast that they are just starting to work on. So this is going to be interesting to see what happens. They are much more complicated because they have a lot of repetition of the DNA inside plant chromosomes. So this will be very, very interesting to see how much progress will be done over the next 5 years with that call that is from UK.
Looking forward to that. And looking forward to the results of all the 3 organisms in your lab.
Yes, me too.
All right. Thank you so much, Bogumil, and thanks everyone else.
Thank you everyone for listening to this.
And have a great rest of your week.
You too. Bye-bye.
Bye.